Now, if this is the first time you're studying glycolysis, I highly recommend you go check out the previous lecture that I did on the introduction of glycolysis and cellular respiration before watching this one. It's roughly about 7 minutes long, but it'll save you hours of future frustration. And if you've already watched the previous video or are just revising glycolysis, then by all means, let's continue. Okay, so I'm going to show you an equation. Now, I don't want you to freak out because it might seem big and scary, but I promise you, once you understand it, you'll say, oh wow, it's not even that big and not scary at all. Okay, so here's the equation. This is net glycolysis. So all this is saying is we're starting from glucose, 2 inorganic phosphate groups, 2 ADP, 2 NAD plus molecules, and we end up with 2 pyruvate, 2 ATP, 2 NADH, 2 protons, and 2 water molecules. And if this does not make sense to you, trust me, at the end of the video, you're gonna totally understand it and it's gonna make so much more sense. So here we have our intestinal cell, and this is the lumen. This is the lumen. And let's say you chug down some Buzz Cola. So what your body's going to do is it'll break it down to glucose and fructose. And so let's say the glucose, let's say this is glucose, enters here, comes here to the intestine, to the intestinal lumen. How does it enter the cell? How is the glucose going to enter the cell? Okay, let's discuss 2 different ways glucose enters a cell. This one is ATP dependent, meaning it requires energy, and this one is called SGLT, stands for sodium glucose transporter. And this transports, as the name suggests, it transports sodium and glucose to the inside of the cell. And next we have ATP independent. So now this is called GLUT, standing for glucose transporters. So there are like around 14 different types, but we're going to focus on 4 major ones. Here we have GLUT1, we have GLUT2, 3, and 4. So we have GLUT1, 2, 3, and 4. So type 1 and type 3, their function is to keep a steady amount of glucose within a certain cell, whereas GLUT2 and GLUT4 have a different function, that is to remove excess amount of glucose from the blood. So GLUT1 and 3 are pretty much found in every single cell, whereas 2 and 4 are found in specific organs. So 2 is found in liver, in pancreas, and kidney. And GLUT4 is present in fat cells and muscle cells. So there's a big difference between 1, 2, 3, and 4, and that big difference is GLUT4 is insulin. It's insulin dependent, meaning it will only work in the presence of insulin, whereas 1, 2, and 3 do not depend on insulin. But GLUT4, which is found on fat and muscle cells, are insulin dependent. Okay, now getting back to our scenario, let's say you chug down that cola and then glucose eventually makes its way down to your intestine. And so here we have our sodium and here we have our glucose molecule. So remember what's this? This is called a SGLT, right? So what this is going to do is it's going to grab sodium and glucose and just shove it in the cell. So next, what the glucose will do is get out of the intestinal cell from here, make its way out, enter the bloodstream, and eventually make its way down to a cell. And in this case, we're going to take a hepatocyte, which is a liver cell. We're going to use that as an example. Okay, before getting into details, let's observe this picture and point out all the basic details without getting technical, right? So first, as we can see, we start with obviously this is the glucose. It is 1, 2, 3, 4, 5, 6. So it's made up of 6 carbon atoms, right? So we start with 6 and we end up with 2 different molecules, which are 3 carbon each, right? So that's the first thing we can notice. And what about this? What is this that seems to be on these molecules? So this P is pretty much phosphate, meaning that a phosphate group is attached to these molecules. All right, let's see. So what else? We see that there's different colored arrows, right? We have arrow that's pink colored and arrow that is blue colored. So the pink, so the pink reddish, it represents a irreversible reaction, meaning it can only go forward. But the blue, on the other hand, is bidirectional. It can go, the molecule can go back and forth. So we'll see there's 1, 2, and 3. There are 3 irreversible reactions that are present in glycolysis. So we're going to go over all the names of all different enzymes. But if you can't remember all these enzymes, commit to memory these 3 enzymes. Okay, so let's begin. So the first thing that happens is that glucose, once it enters the cell, right? Once it has entered the cell, the first thing it's going to do is attach a phosphate group to this molecule, making it glucose 6-phosphate. Now the enzyme is hexokinase or glucokinase. Now this is very important. It's a high-yield enzyme, so I'm gonna have a whole different lecture covering just this topic. So this phosphate group, where does that come from? It comes from ATP. So ATP is converted to ADP, and the phosphate group goes from ATP, it goes to glucose, making it glucose 6-phosphate. Next, we have glucose 6-phosphate turning into fructose 6-phosphate. And this is done by phosphoglucose isomerase. Next, we have fructose 6-phosphate turning into fructose 1,1,6-bisphosphate, meaning that there are now there are 2 phosphate groups. And guess where that phosphate comes from? It comes from ATP, which then turns to ADP. So now remember, this is done by phosphofructokinase 1, and this is an irreversible reaction. So meaning it will be very well regulated. So then the question is, what regulates this? What regulates the PFK1 or phosphofructokinase 1 enzyme? So, okay, think about it. The job of glycolysis is to create energy, right? So if there's a good amount of energy present, we wouldn't want to continue with glycolysis. Now, what are some of the indicators that indicate there's high energy? Well, ATP, right? So if there's high amount of ATP in the cell, it will deactivate phosphofructokinase 1. And other thing that can inhibit it's something called citrate. So citrate also indicates that the cell has good amount of energy. So these 2 will inhibit it. So what about what activates it? What do you think activates it? Well, what's an indicator for there being low energy? Well, we have the molecule AMP. So AMP will indicate there's low energy, and that will activate this enzyme, right? But there's another molecule that activates phosphofructokinase 1, and that is called fructose 2,6-bisphosphate. It's fructose 2,6-bisphosphate. That's bisphosphate. I know, it's great handwriting, but anyways, so fructose 2,6-bisphosphate comes from fructose 6-phosphate, and this is done by an enzyme called PFK2. So here we have PFK1 that turns fructose 6-phosphate to fructose 1,6-bisphosphate, and we have PFK2 which turns fructose 6-phosphate to fructose 2,6-bisphosphate. And this right here is a very, very strong activator for phosphofructokinase 1. And this is the most potent activator of this enzyme, even in the presence of ATP. Even let's say there's ATP, high amount of ATP present and high amount of fructose 2,6 bisphosphate present, the cell will prioritize this molecule right here. So next we have fructose 1,6 bisphosphate converting to glyceraldehyde 3-phosphate and dihydroacetone phosphate, also known as DHAP and G3P, right? So now it's gone from a 6-carbon to 2-3 carbon molecules, and both of these are converted by the enzyme called aldose. But see, there's a problem here. G3P can easily continue on to glycolysis, but DHAP cannot. So in order for DHAP to continue to glycolysis, it has to be converted to G3P. And the enzyme that does that is triose phosphate isomerase. So far, this phase from here to here is considered investment. This is considered investment phase. So from the next stage onwards, it will be considered a yield phase because we're getting energy in return. Here, we invested energy. Right here, we invested ATP right here. And ATP, we invested it right here. So what's the next step? The next step is converting glyceraldehyde 3-phosphate, G3P, to 1,3-bisphosphoglycerate. Now, this is an important and very interesting reaction. This is something called an oxidation reduction reaction. So what does oxidation reduction reaction even mean? It basically means, let's say we have a molecule here and a molecule here. So one of the molecules has an electron and the other one doesn't. So the molecule that donates it, it's called oxidation, and the one that receives it is called reduction. So this electron basically ends up right here, and this is known as the oxidation reduction reaction. How do you remember which one donates and which one receives? There's a mnemonic called oil rig. So the O here stands for oxidation, and L is for loss, and R is for reduction, and G is for gain. As simple as that. So this molecule, glyceraldehyde 3-phosphate, specifically the aldehyde group in this molecule, donates an electron, and it donates it to something known as NAD+. So the NAD+ converts to NADH. Now keep this in mind. This is happening to both the molecules here. And you see there's addition of this phosphate group. Where is this coming from? Does it come from an ATP? Not really. It comes from, say there's random phosphate groups just lying around in cell. This reaction that happens here, it also adds this random phosphate group that's in the cell to this molecule, turning into 1,3-bisphosphoglycerate. And during this reaction, we also get release of a proton. Remember, we have release of 2 protons and 2 NAD molecules converting to NADH. Now here's something important. The molecule NAD+ is crucial. It's crucial for glycolysis. Without NAD+, glycolysis cannot continue. And the cell has limited amount of NAD present in the cell. Now let's connect this NADH that will be made here. This NADH will then go ahead and make its way to the electron transport chain. I want you to make this association, and we're going to go into details regarding the electron transport chain. But for now, just know that NADH that is made here will then go ahead and make its way to electron transport chain, where it will donate an electron to this right here. So coming back to glycolysis, what's next? Next we have 1,3-bis phosphoglycerate that turns to 3-phosphoglycerate. Now look at what's happening here. We're getting rid of a phosphate group. Now guess where that's going? That's going to a ATP molecule. So now we're taking ADP and converting into ATP, and that is done by phosphoglycerate kinase. Keep that in mind. It's happening to both of these. So the 2 ATP molecules that we invested in this in the investment phase, look at it, we've already gotten it back. Next we have the conversion of 3-phosphoglycerate to 2-phosphoglycerate. So here the phosphate group just moves from the 3rd position to the 2nd position. It's done by phosphoglycerate mutase. Next we have dehydration of the molecule. So we have 2-phosphoglycerate turning into phosphoenol pyruvate, and in this we release some water, release 2 molecules of water right here. And finally we have the conversion of phosphoenol pyruvate to pyruvate. Now take a look at this. Take a look at the phosphate group. It's gone, right? So what do you think that means? That just means we converted another ADP back to ATP. And again, it happens on both sides. We take the ADP and convert it to ATP. And this is done here by pyruvate kinase. And here's another interesting fact. You see this molecule here, fructose 1,6-bis phosphate. What this does is, this can also come here and activate. This can activate the pyruvate kinase. And on the other hand, we have ATP here. So ATP here can come and deactivate the pyruvate kinase. Okay, now coming back to our equation that we started with. Let's take a look at it again. So here we have 2 pyruvate. That is this one right here. First, second pyruvate. We have our 2 ATP molecules. Now, hang on a minute. I thought we made 4 ATP molecules. Why just it 2 here? Because remember, this is a net reaction of glycolysis. So let's count our ATP molecules again. So here we have our first one right here, ADP to ATP. That's 1. This is 2, and the third one is right here, ADP to ATP. Another one right here. Look at that. We have 1, 2, 3, 4. So 4 ATP molecules, and we invested 2. This is our first investment, and this is our second investment. So total we have 4 minus 2. I'm sure you guys can do simple maths. That's just 2. So that's what this number represents here, the 2 ATP. And then we have 2 NADH. Now where did that happen? That happened right here. Here we took our NAD plus and converted it to NADH. Happened on both sides. That's what this is for. And the 2 protons also happened right here and right here. We had our 2 protons and the 2 water molecules that were dehydrated right here. I mean, this molecule was dehydrated and we got 2 water molecules out of it. And this is what we started with. Here we have a glucose, or to this glucose molecule, the 2 inorganic phosphate groups. Remember, that happened right here when we added a phosphate group right here. Boom, boom. This 2 ADP, talking about the net, here, and then 2 NAD, which is right here. Here's first one and second one. That's it. It's as simple as that. So we're finally done with glycolysis now, and in our next video we're gonna go over anaerobic versus aerobic glycolysis. And the good thing is we've already covered all the hard parts, so that video will be much more easier than this one. Thank you very much for watching this video, and if you found it helpful, make sure to like and subscribe and share with someone who might be struggling in this topic. Go make their day.